and welcome to the University of Alberta's Speakers Bureau online presentations. My name is Anastasia Lim. I'm Executive Director of Community Relations here at the U of A. A couple of weeks ago, we had the great pleasure of having Dr. David Evans and Dr. Mary Hitt speak to us about what to consider during stage 2 reopening of our economy post-COVID-19. I will introduce our two professors and then turn the floor over to them. Enjoy your learning journey. Dr. David Evans is a virologist with diverse interest in poxvirology biology, as well as former research administrator. He is considered an international leader in the study of poxvirology, with special expertise in virus recombination, antiviral drugs, and replication. Dr. Evans' activities also helped develop the University of Alberta's focus on virus research. Dr. Mary Hitt is an associate professor in the Department of Oncology at the University of Alberta. Since the 1990s, she has investigated ways to modify the viruses so that they target and kill cancer cells. About 8 years ago, Dr. Hitt and Dr. Evans joined forces to develop a virus that they hope to test in a clinical trial for bladder cancer in the near future. And with that, I turn it over to Dr. Evans and Dr. Hitt.
Good morning, everyone.
Good morning. Do you want to start the presentation?
Should we start the presentation? All right. One thing I just wanted to point out, we are not socially distancing ourselves because we're actually married to each other. So in case you were worried. So I'll start sharing the screen here.
Okay.
How's that? Can everyone see that?
Yes, we can see it.
Okay. So let's see, I think I'll start off.
No, just maybe click on that.
This one?
Just
click.
It's down there.
Oh, okay. All right. All right. So thank you, Anastasia, for the nice introduction. So I don't need to really say too much of what is on this slide. I just wanted to point out that I was actually part of a team at McMaster University that developed a SARS vaccine back in 2003. So I do have a little bit of experience with working with vaccines and coronaviruses, but not really a lot. Mostly what we've been doing is the development of viruses to kill cancer cells. But I have been keeping up on the COVID-19 literature, and so that's why I'm able to participate in this talk. So I don't know how many people have already seen this website before, but this is a website that's set up by the Johns Hopkins University of Medicine, and it shows all of the total number of cases that have been detected or reported globally. And so it's a really nice visual to show that the COVID-19 is actually a pandemic. You can see how dark all the red spots are where there have been different cases. So it's a really nice website to browse if you're interested in statistics. The website's up here, but I think this talk will be available afterwards, so you don't have to try to write down all the websites that are here. So this site is updated a couple of times a day, I think. So it's a really good way to look at total cases of coronavirus. But there's another website we'll get to here that's set up by CTV News, and this one is interactive, and I actually like this one better because you can follow the number of cases that are reported on a daily basis, and in this case we're looking at cases that have been averaged over, new cases over a 7-day period, and it's per 100,000 population. And on this website, you can pick any 4 countries you want to see, clicking on them down here. And what I've shown here is Canada is right here. So number of cases per day, and you can see that cases are really going down a lot. We've got the United Kingdom, it's going down. The US, not going down so much as you might like, since there are neighbors to the south. But I also wanted to show you Brazil. So Brazil and other places in South America and in India actually are still really on the upswing of reported cases. So even though things seem really good here in Alberta, and it is really good here in Alberta, that's not the case globally. And so you still have to consider some of the restrictions that have to do with the fact that there are still a lot of people in the US and certainly other people internationally that have COVID infections. So also on this website, you can look at different provinces, which is also very interesting. So again, this is new cases averaged over 7 days, and it's in per 100,000 population. We have Alberta's in the red here, Ontario's black, and Quebec is this orangey gold color here. So you can see that things have been really bad in Quebec, but it's definitely getting a lot better. Alberta had a big blip month and a half ago, but now it's gone down really, really well. And I think the fact that this has gone down really well is due to so many people following the restrictions and not spreading the virus all over the place. So good job, everybody. This website also lets you look at deaths, and I didn't print that out. But Alberta's really not had a lot of COVID deaths at all. It's been really low all along this. And I think that's really thanks to Alberta Health Services and the great job that they've done in treating people who are COVID positive. So as of yesterday, there were over 300,000 tests done in Alberta. Over 7,000 of them were positive cases, and we've had 151 deaths in Alberta, which spread over a couple months, I think is very commendable considering what it's like in other places. So right now we have 356 active cases, 46 people in the hospital, well, yesterday anyway, and 6 of them are in intensive care units. So that's way below the 1,000 acute care beds that have been set aside for the COVID infected patients. And because these numbers are so low, and it's been staying low for a while, we had a little blip just recently, but it's still fairly low levels. So that's why the Alberta government has decided to loosen up the restrictions. And if I can get to this. So stage 1, which is where we are today, and it's ending tomorrow, because Friday we start the new phase. Stage 1, there are retail businesses that are open, hair salons, cafes, restaurants. Some surgeries are allowed now, museums and art galleries, daycares, day camps. Universities have courses online, and some places of worship and funeral services have all been allowed to open, but there's a lot of restrictions on what they have to do in order to open. And if you are interested in more details, you could look at this website that's up here. But one of the big important things is that this 2-meter physical distancing requirement is really, really important, and that's what's driven a lot of the restrictions that are going on here. Likewise, for stage 2, which starts on Friday, the 2-meter physical distancing requirement is still in place, and that also is driving the restrictions that are going on here. So I didn't list everything that's opening with stage 2, but I hit some of the highlights. So libraries were going to open up, and I'm very excited about that, but now I have to find the library book that I checked out in March. Personal services are open, cinemas, theaters, team sports, pools will be open, bingo halls, casinos, and campgrounds is also wonderful. But these all still have restrictions, mainly to do with this physical distancing problem. So events and gatherings can be a lot larger in stage 2 than they were in stage 1, and you can check out the website to see how big and which events and gatherings we're talking about here. But there's still some things that aren't approved in stage 2 that won't be approved until stage 3. So not approved are singing concerts, because singing carries a high risk of transmission. Major festivals and concerts and conferences, because a lot of times people are mingling at those events. And then sporting events are not open, I think, due to just how densely packed people are, and they get excited and shout and things that are issues. Nightclubs, non-essential travel outside of the province is still not recommended during stage 2. And that comes from those statistics that I showed you before, where there's still quite a few cases south of the border, and in other countries there's quite a bit, and even in other provinces there's quite a bit. And so it's really just trying to keep the numbers down on new cases. All right, so last couple of slides that I talk about, I just wanted to go over a little bit of the science behind these restrictions. So first, COVID-19 is spread person to person through contact of mucous membranes. So that's eyes, nose, and mouth, with secretions from an infected person. So this could be direct, so they could be breathing right on you, or it could be through heavily contaminated surfaces. So it doesn't have to be directly from a person, but it has to be from it. To catch it from a surface, there would have to be a lot of virus on the surface, and it would have to be live virus. And I think Dr. Evans will talk a little bit more about that when he talks. So the best protection to avoid this person-to-person spread is to prevent contact with droplets from potentially infected people. So stay 2 meters apart or use a mask. So the mask is mostly important for preventing you from spreading your droplets onto everyone else. If you have a mask on and you're standing 2 feet from someone that coughs and they're infected, then the mask is not a lot of protection. But it does keep someone else, it keeps people who are infected from spreading as much as they would if they didn't have a mask on. So the other thing, important, wash your hands often to prevent the transfer of virus from infectious people or from contaminated surfaces to your eyes or your nose or your mouth. And then finally, to disinfect surfaces. And the main point with the washing hands and disinfecting surfaces is that the risk of catching this viral infection goes up with the number of people that have touched the same surface that you touched, because the more people that might have touched it, the more likely one of those people might be infectious and, well, know it or not know it, but one of them might be infectious. So the fewer people touch it, the less likelihood there is that you're going to catch anything from the surface. Okay, a little bit more on the science is a person is more likely to spread the virus. So it's called SARS-CoV-2 is the virus that causes COVID-19. If the people have symptoms, the symptomatic individuals have more secretions, and those secretions have higher viral loads than people that are asymptomatic or pre-symptomatic. The peak virus load occurs at symptom onset or slightly after. So that's why it's really important to self-isolate for 10 days if you have any symptoms that sound like the symptoms for COVID-19, because that's when you're, right at the very beginning is when you're most likely to be able to spread it. The virus might also be shed 1 or 2 days before symptom onset. There's a little bit less data available on this, but it does look like a little bit before the symptoms start, you might be infectious. So that's why it's important to wear a mask all the time if you can't distance, because it's really hard to know if you're going to get symptoms in a couple of days if you don't have any yet. So always wear a mask if you're going to be coming up close to someone else and you're out in a public space. Obviously, you don't have to do that at home. There are asymptomatic individuals, those are the people that don't have symptoms. And it's possible for them to be positive for the virus, but it's not very common. So just a couple of days ago, Dr. Hinshaw was on Global News and said that there are, of the almost 15,000 asymptomatic Albertans that were tested for virus, only 6 of them were positive. So that's really not, 6 out of 15,000 is really not very many. And of those 6, it's not really clear how well these positive individuals can transmit virus to other people. And the test that you use, that everyone's using, is a test to look for RNA that's part of the virus particle. So it will tell you whether or not, the test will tell you whether or not the virus is there, but what it doesn't tell you is whether the virus is actually infectious, whether it's a live virus or if it's a dead virus. And it's not clear with these asymptomatic people if it's dead virus that they've got, if it's live virus, or if it's just such low levels that they're not really spreading it very much. So another website that I found really useful to look at is one that the Alberta Health Services has made, and it's really was designed to educate healthcare workers, but I think there's a lot of information that you might find interesting in that. So if you look at this website and scroll all the way to the bottom, there are some webinars on the 5 key messages, and they basically have the same messages, but different clinicians explaining why those messages are important. All right, so I'll hand it over to, oops, I think I have one more actually. Maybe 2 more.
Nope.
Nope, that was it.
Just me.
Okay.
All right.
There you go.
All right, thank you, Mary. So again, Anastasia gave a nice introduction, so I won't go over this in too great a detail. What I wanted to do in the few minutes I have left to talk is talk a little bit about some of the work we've been doing in my own lab as we've kind of pivoted away from the main lab research on poxviruses to studying COVID-19. And so I'm going to give you just a very sort of brief overview of some of the work we've been doing that serves to illustrate some of the points that Dr. Hitt was identifying and which have been stressed by the health authorities, which are why we have these constraints on Albertans' behavior. So as was introduced, I mean, Dr. Hitt and I have been working in particular on oncolytic viruses, and we have a particular interest in bladder cancer. But the COVID-19 response, what we've been looking at are sort of four different things. One of the things we've been looking at is how one can go about inactivating this virus. There's been a particular interest in trying to figure out how to do that with rather resource-limited places. Alberta's lucky; we have a lot of advanced technologies, but there are many parts of the world which don't have access to those technologies. We're also looking at some of the mechanisms of spread, and that's been quite informative. I won't really talk about the neutralizing antibody work. We're just starting that in collaboration with the Canadian Blood Service. And then finally, I'll just give you a real flavor, just a brief flavor of some of the work we're doing on the development of a vaccine based on our expertise in poxviruses. So, so what are some of the things we've learned about SARS coronavirus 2? Whoops, sorry, I go back. Just first thing I want to point out to you is that the challenge with working with this virus is that it's considered a BSL-3 agent. That is, it requires special protection to work with it. We're very fortunate to have built over the last decade and been operating BSL-3 biocontainment laboratory. And this picture here shows one of my colleagues working in that laboratory. So what Dr. Nagat is doing there is growing some virus. These are the flask we use to grow the virus. But you'll notice the protection he wears to avoid himself getting infected. This is a special air containment unit. And when he leaves that facility, he actually sprays himself down with decon to remove any virus that might have landed on the outside. And this facility has actually got special air handling conditions which ensure that nothing can get out and everything is sterilized before it leaves. So using this resource, we've been doing a number of experiments to try and determine some ways in which you can kill this virus. Virologists don't like the word live and dead to describe viruses because viruses aren't really alive in the first place. We usually talk about infectious and non-infectious. So, but I'll use the word dead as a shorthand here. So one of the things we've learned is the virus is very fragile. There's a lot of things that actually will kill it. And one of the things that's actually very simple that works quite effectively is simple heat, dry heat. 15 minutes at 65 to 75 degrees will inactivate the virus. That's why if you wash your, say a mask in soapy or clothing in soapy hot water, you can inactivate the virus, especially if you give it through a dry cycle in your dryer. We have noticed in some of our experiments with light that it's actually inactivated by light, and there's actually been publications showing that bright sunlight is a very effective way of killing this virus. So if you go outdoors, I would not worry too much about things being on park benches. Bright sunlight will be killing that. One of the reasons for that is sunlight actually contains ultraviolet light, and we've also been working with a company in Alberta to test the use of ultraviolet light as a sterilizing technique, and it works really, really well, I can assure you. Not a common thing you have at home though, but it is the reason why sunlight is such a good killer of the virus. You're no doubt well aware that common cleaners, your Lysols and stuff, those are very effective. The reason for that is this virus is surrounded by a fatty membrane, and not unlike, you know, you've probably seen a sink full of cold dishwater where the fat is kind of congealed on the surface in a sort of layer. If you add a few drops of liquid soap to it, you'll see that layer disappear on you. And that's exactly the same effect that common detergent-based cleaners have on the lipid layer of these viruses. They're very effective at killing it. Also 70% alcohol works instantaneously, and 1% bleach is also a very good thing to sterilize this virus. And there are a number of websites like Health Canada, which will summarize some of the approved cleaners that work really well. This just gives you kind of a flavor of the sort of experiment we do. This shows heat being used to inactivate the virus. And so what you're seeing on this plot here is we had samples of virus stored either at room temperature or in a little heater block at 65 degrees. And what you see on this axis is we started with a little over 1 million viruses in our spot of virus, about 2 million viruses. And over time, we took samples out and we measured how much virus activity was left. And what you can see that in 15 minutes at 65 degrees, we destroyed nearly it dropped the titer of viruses, we would say about 10,000-fold. And within 30 minutes, which you can't show on here because our limit of detection is here, there's no detectable virus. Meanwhile, at room temperature, the virus seems to be pretty stable actually in those surfaces, just illustrating how effective heat is for doing this. So another thing we've been spending a lot of our efforts on is looking at the mechanism of spread. And we're particularly interested, as Dr. Hitt pointed out, on the issue of measuring infectious virus, live virus, not the nucleic acids in the virus, which is the basis of all the diagnostic tests. And this is how virologists would look for the presence of viruses. These are what are called plaques. So here's a dish which had some cells in it, some mammalian cells in it. And it's been stained at the end of the experiment with a dye, crystal violet, so that you can actually see where all the cells are. And when we started this experiment, 3 days before this was done, a sample of virus was plopped down on it. And every place where a virus landed, it started to cause an infection of the cells. And as it grew outwards, it killed the cells. And so when we stained it at the end of 3 days, you see places where the virus is killed and caused a ring. And each one of those plaques reflects the presence of a single infectious particle at the beginning of the experiment. So we can count how many viruses there are doing this. And we've been working with John Connolly, as a doctor down at the University of Calgary, who treats a lot of patients with COVID-19. And it's been quite interesting. One of the things that and some of this we are confirming the stuff that's in the literature. The first thing is that you can, in fact, detect a lot of infectious virus in saliva and mucus from a person who has got COVID-19. That's how it spreads, basically. But interestingly, what we discovered, and other laboratories are discovering the same thing, about a week after symptoms first appear, detecting infectious or live virus becomes very, very difficult to do. If you use a nucleic acid test, you can still see it there, but it doesn't seem to be infectious anymore. And that's presumably due to some kind of immune response, which is starting to inactivate the virus. The other thing that has been really quite interesting, and the thing we started out to do, was it's actually quite difficult to pick up infectious virus on surfaces in those where those hospitalized patients are. And we think that reflects, various factors, but it shows that unless someone really heavily contaminates a surface, and we have seen surfaces like that, it's very difficult to pick up virus from the surfaces. So as Dr. Hitt was mentioning at the beginning, it's the things that are touched most heavily by people, and obviously people who would be quite ill, it's where you're going to most likely pick up the virus if you're going to spread it by that route. So some of the things that, just to summarize some of those things, common cleaning methods will inactivate this virus, and even sunlight. As your grandmother used to tell us, sunlight on things outdoors is a good way to clean them and sterilize them. It's difficult to transmit the virus from surface, although beware, if they're heavily contaminated, you could still do that. Our data, again, other data that's coming out, is the infection is most infectious very early in infection. And this declines as the disease progresses. So sort of the thing that's becoming sort of widely accepted now is that most of the spread is through droplets and aerosols. And you may have seen this very famous picture taken about 60 years ago at MIT showing someone sneezing. and all the spray of material that comes out of a person doing that. That's an extreme case, but it's these particles, which is what is particularly important as a route for spreading this kind of virus and other viruses as well, like flu, for example. So talking to some extent, and certainly singing, will produce small amounts of aerosols, but obviously sneezing, coughing, and spitting are particularly good at doing that. So hand washing is still very important. I'm not just discounting that. You really don't know whether a surface is being contaminated by someone with the disease, but the focus on masks and protecting yourself from the getting virus on you, your mucous membranes, is also very, very critical. The last thing I'll just tell, I won't go into this in any great detail, but I know there are probably some science buffs on the talk today. Just tell you a little bit about our own vaccine efforts that are going on in my laboratory. So what I study is vaccinia virus, and here's a sort of model of the particle. It's a big virus. Some of you of a certain age may have been vaccinated against smallpox. We haven't done that for 50 years, but you may have a big scar on your arm, and that was vaccinia virus that's used to vaccinate against smallpox. It was used to eradicate smallpox. And we study vaccinia virus. And so what we're interested in is whether it can be used as a vector to make a COVID-2 vaccine. So the way we did this is that this is a map of the genome of COVID virus. Okay, it's about 29,900 nucleotides long. It encodes a whole group of different proteins. Some of them make virus, some are in the capsid of the virus. And so what we did was we used gene synthesis to copy bits of this genome. For example, this is what's called the spike protein, the S protein. And in that picture that Dr. Hitt showed you, where it's a famous picture where you see the red knobby structures on the surface of the virus, that's the spike protein. It's important for binding to a cell. And so we cloned these things into vaccinia virus. We stuck the genes for them into the DNA of the virus. As an example, we know, for example, that this recombinant version of the S spike protein can be expressed very well by vaccinia. And so what we're now testing is, just last week, we did some vaccinations in some mice, and we're looking to see whether they produce an immune response to the recombinant protein. And we have some other proteins as well that we're testing. If that all looks promising, we're working with a company in the United States who's sponsoring some of the work, and they're hoping to move very aggressively into some clinical trials if this is successful. I always get the question from people, well, how long will this take? I was bullied once by a reporter into saying maybe a year and a half, but I think that's optimistic. This kind of work takes a long time, and realistically, it's at least a couple of years, maybe more, before one could have enough safety data to justify vaccinating significant numbers of people, but we'll see. So with that, I'll say thank you for listening to us, and we'll happily answer any questions.
All right, thank you very much for your presentations. That was really interesting. And yes, some of us science geeks right here, we're really interested in hearing more about the structure of the virus and everything, so that's wonderful. I throw the floor open to any questions that anyone may have. Anyone have a hand up or anything? If not, I can ask somebody questions that were submitted in advance through the registration. Well, while everyone's warming up, I'm going to go ahead and ask one of the questions then that came in from the registration. So we talked a little bit about cleaning surfaces and things like that with Lysols and such, but how do we clean fresh produce?
So there's a lot of different ways that you could do it. So one is not to worry too much if you're buying produce that's already been bagged, but if you are like me and you like to pick it up and look at it and then put it in the bag, then so is everybody else. It's gone through the grocery store that day. So it's useful to clean it off just because you don't know. So soapy water, like dishwater, is fine. I think someone had written in about using peroxide. I think peroxide is probably okay. I don't know if peroxide does anything to the taste of the vegetables or not. But I think especially things that vegetables or produce where you're eating the outside of it, you do want to make sure that it's been cleaned off. I mean, you would probably anyway, you wouldn't eat an apple that you hadn't washed off if you bought it from the grocery store, but same kind of thing. So you don't have to do anything super special. Soapy water is fine.
Okay, soapy water's fine. Okay, great, thank you.
And of course, if it's going to get cooked, it's going to be sterilized.
Yes,
Yeah.
absolutely. So on that note about cooked, for example, those who might like foods like beef tartare or sushi, someone's handling that food as well. Is there a fear in that case in terms of what is safe to eat, and maybe we shouldn't be eating sushi for the next while, anything like that we should be cautious about?
I mean, my feeling is it all has to do with risk. And the more people that have their hands on something, the greater the risk is that it's contaminated. But if you get, say, steak tartare from the grocery store, there's one person really who's ground the meat up. So the chances of that one person being infectious are maybe not so great. But definitely there's more of a risk to eat that than to fry it up and have it cooked. And same thing with sushi, I guess, is the fewer hands that are on it, the lower the risk is that there's virus on it as well.
And the thing to also remember is that it's not just COVID-19 that you have to worry about when preparing those kind of raw meats. The butchers and stuff are very cognizant of the bacterial contamination, the E. coli and stuff. So the procedures that are adhered to prepare that in the first place would protect you from COVID-19 as well as they would protect you from any other pathogen that might be on them. And there's actually there's more I'd worry about than I would COVID-19, like E. coli,
with raw
meat, with E. coli 0157, for example.
Right. Okay, thank you. A question just came in through the chat from Lynn, and it says, are you concerned about working with the American company in terms of keeping resulting vaccines just for the US? Are scientists working together across the world?
That's an interesting question. It's very hard to say. We're actually making several different kinds of vaccines, and we're making one with a Canadian focus with our own vectors, and we're making some other ones for this American company. I'm doing that because there are some particular advantages, I think, of one vector over the other, which I won't go into here. It's very hard to say down the road what the rules would be on the distribution of these viruses. An American company, of course, would always focus on getting US Food and Drug Administration approval in the United States. So to bring a vaccine approved by FDA in the US back to Canada would require the Health Canada's approval. And one would have to then look into what are the process for getting their approval. People are collaborating on this. There's a lot of interest in trying to ensure that these things can be shared widely. But at the end of the day, companies seem to be the people willing to put money forward, and significant sums of money forward, to make these things happen. And I think you have to respect the fact that they need to get their money back in the long run. When we don't have the large sums of money that are needed to develop these vaccines in Canada with Canadian government funding. So hopefully that will change. But right now, relying on companies with their resources to do this is what we have to do.
Yeah. Also, I don't think there's a huge risk of any countries hoarding the virus. I think they would make what they needed for themselves first, but after that, they would allow it to be used by others. I don't think there's a problem with preventing other people from getting it just because. So I think in that sense, it's a global effort to come up with vaccines.
Okay. We've got a couple of other questions. One that came from registration that ties back to the vaccine as well is, are we anticipating, should a vaccine become available, that it's its own standalone vaccine, or would it be something that might be rolled into the regular flu vaccine as well?
Yeah, that's interesting. And Dr. Hitt and I talked about that a little earlier. The thing is that there are different methods of delivering different kinds of vaccines. So for example, the flu vaccine is typically an inactivated vaccine that's injected into your skin. Now, depending on what other type of vaccine gets improved, you may require a different delivery mechanism. So you can't mix two different ways of delivering a vaccine. It's just not very practical. And then you had another point, Mary, about
well, some of the flu vaccine, I think, is grown on eggs or something, right? So I mean, there could be people that are allergic to eggs, so they can't take that. So then you wouldn't want to have it mixed together for them. But you might be able to go to the doctor and get a jab in one arm with one thing and a jab in the other arm with the other. So you might still get treated at the same time. It just wouldn't be in the same vial.
Right. Okay, great. Thank you. We have a number of other questions that came in. So there's a question, it's kind of a two-parter. So one is, can you comment on whether we should travel in Canada? And then secondly, do the cloth masks that we use need to be washed and dried in washing machine and dryer? So I think that speaks to the heat aspect that we might have been talking about earlier.
Why did I take the travel and you could do the mask?
Sure.
With respect to travel, I think it depends where you're traveling to. To be quite honest, with what's still going on in Quebec, I would be hesitant to go to Montreal right now. Whereas, you know, I think if you were going to the mountain parks in BC or some of the beautiful parts of Saskatchewan, I would be far less concerned about it. And as long as you are maintaining your social distancing while you're doing that, I don't see there is going to be a problem. Although I gather some people with Alberta plates had some rude notes put on their cars recently when they were traveling to BC. So whether we're welcome there is another question, but I don't think there's any great risk of traveling to those, to at least locally anyway.
Mm-hmm. Yeah. So, and as far as the masks go, you could wash the mask out in a cloth mask out in the sink. If you use really hot water, hot soapy water, it would be okay. And you could, this time of year, you could hang it outside to dry, and that would probably be fine. So it doesn't have to be in a washer and a dryer, but it does have to be washed somewhere. You don't want to keep using the same mask again and again because it's dirty. Yeah.
Right. Okay, great. Thank you. There's another question from Debbie. Do we know how much virus will change a person from asymptomatic to symptomatic? So what's that threshold that's going to push you over to really being sick with COVID?
That's a really good question. And to be honest, nobody knows. The only way to really do that, to figure that out, is actually take a series of volunteers and expose them to different doses of virus and see who gets the disease. Of course, for ethical reasons, that would be, we're not going to be doing that. We don't really know. And the other thing about viruses that needs to be remembered is that oftentimes there's, what's called the particle to PFU ratio. The fraction of virus which is infected versus the fraction of virus which is dead varies a lot from virus to virus, and will of course vary, of course, over an infection. And that can confound efforts to try and figure out, well, how much virus is there? How much infection did it cause? And then finally, people vary a lot in their sensitivity to viruses. One of the things that's being debated fiercely in the scientific community is that, you do seem to have people who are exposed to the virus and you can detect the virus, but they really don't show any symptoms of it. They don't really get very sick. And one suggestion has been is that there are lots of different kinds of coronaviruses out there, and some very common cold-like symptoms that people get are caused by other kinds of human coronaviruses. So there's a suspicion that it may be that people who've been exposed to other kinds of coronaviruses are partially protected against this one. And so, that just depends on your history of getting colds. And so it may be that an individual who was exposed to another coronavirus 10 years ago would be much more resistant to infection by the virus than someone who had never seen it.
Okay. I also have a question here about Just wondering if you have any suggestions for cleaning keyboards and mice for computers.
Yeah, I mean, I think if you use isopropanol, that works pretty well. Like 70% isopropanol is fine. You just don't flood it, or you'll have problems with your mouse functioning and your keyboard functioning. But you could spray it on a cloth and then wipe it down. Should be okay. I mean, I guess the other question is how many people are using the keyboard, and is it a keyboard? Is it a pin pad at the grocery store that lots of people have their hands on, or is it one in your house and there's just 2 or 3 people using it? So, I mean, you can put, if there's a lot of people using it, you can get some kind of like a plastic sleeve that goes over the top of the keyboard that's washable. And then you can spray it as much as you like with isopropanol. It'd be fine.
Great. Okay. I also have Ralph who has his hand up. Can we unmute Ralph's microphone, please, and let him ask his question? Ralph, are you there?
I'm not sure if I'm the right Ralph that you referred to.
Yes, yes, you are. Thank you, Ralph.
But I do have, I notice on chat an interesting question that relates to myself, and that's the question with respect to indoor swimming pools, and whether you could comment on the virus life in the indoor pool environment regarding actual swimming, sauna, and in particular the locker rooms.
Thank you, Ralph.
Yeah, no, that's a very good question. I mean, one of the things that has been observed is 1% bleach is a very effective inactivator of the virus. So I don't know what the levels of chlorine are. I mean, main reason bleach works is the chlorine in it. I don't know what the levels of chlorine are in pools, but they're usually pretty high, and they're actually designed to kill things much tougher than coronaviruses, bacteria, and so on. So I would imagine that any pool that's got a reasonably, well-maintained level of chlorine in it is going to be pretty much free of a coronavirus.
Yeah, the locker room's the other issue.
The locker room is another question, yeah.
I would go with the physical distancing. So as long as people aren't all crowding into the same space, if they're all spread out in the locker room, it should be okay. Might be worth, if you were using a locker that's there, just wiping it down a little bit before you put your hands all over it.
Right.
It should be okay. And saunas, I don't know about a sauna. That's a dry sauna or wet sauna. So dry sauna is probably okay. Wet sauna, I don't know.
Well, the virus does inactivate somewhat more rapidly in a humid environment than in a dry environment.
Inactivate?
Yes, it's more sensitive if it's humid heat than dry heat. So yeah, but unless you want us to come over and put some in your sauna, I don't think we have an easy way of testing that. I'm sorry.
Okay, I've got a number of questions here as well. Debbie asks, how has the concentration of research on the coronavirus after research in other areas? So I think the question is, how much of the coronavirus research is occupying the overall research umbrella in virus research?
Well, I've seen a lot of labs drop. Well, first of all, I should just, for people maybe not familiar with it, CIHR, the Canadian Institute for Health Research, has actually produced some very special funding for COVID-19 research, quite a bit of money actually. Similar support has come from the Natural Sciences and Engineering Research Council, and there's some other funding as well, quite a bit of it. So it has led to a number of labs sort of pivoting and starting to pursue other kinds of research. So for example, some of our work is supported by the vaccine work is supported by a CIHR grant. I think it has, in the medical research field, it's hard to put a number on it, but there are, I don't know, maybe 10, 20% of labs have sort of taken on COVID-19 research in a big way. Although not very many are doing it like we are because of the requirement for the BSL-3 work, the high containment work. But what you do have, for example, is laboratories that study proteases, like some colleagues of ours in the biochemistry department. Proteases are, the COVID-19 encodes a protease, or a couple of them actually, and these enzymes are important for chopping up that virus genome, all the proteins in it, and making more virus. And some very effective antiviral drugs have been developed to target proteases, some of the best HIV drugs, for example, the new hepatitis C drugs. So you have people like this taking their expertise in proteases and now looking at whether you can repurpose other small molecule drugs to inhibit the COVID-19 proteases. So I'm not sure I'm really answering the question very well, but that's an example of a repositioning of a laboratory where their expertise is very valuable in helping to devise some new drugs for treating this virus. And it's probably in the biomedical research area, I don't know, 10, 20% of laboratories have got some reapplied research that involves COVID-19. Would you guess that? Yeah, I don't know exactly.
Okay, thank you. I have a question from Louise and then also one from the Preville family. So from Louise, back to some of the reopening and relaunch categories. Libraries will be opening soon. What will we need to be aware of when we take a pile of books back and take out new ones? Because spraying them down and washing in soapy water may not be the right way to do it then, but what would your suggestions be then as we go back and look at paper materials, even in the office? So we go back to the office and look at manuals or photocopy equipment, things like that. So what should we be aware of?
Well, I mean, the chances of the virus being there aren't really high, but one thing that you can do if you check books out of the library, if you come home and wash your hand, put the books someplace and leave them for a few days and then wash your hands, and in a few days any virus that would be there is inactivated. So all you have to do is be patient and wait a little while and the virus will be gone. So I think that would be what I plan to do, is just wash hands before I bring things in, wash hands when I bring it home, and then just leave it for a little while before I read things. I think that would probably cover it. Yeah, and same thing, I'll be really surprised if libraries don't have hand sanitizers right inside the door so that you can clean your hands as you go in and clean them as you come back, so that even someone coming in, if they they weren't really infectious, they were, say, the pre-symptomatic phase, I think washing their hands coming in would do a lot to keep the virus down, especially people wearing masks at the same time.
Right.
There's been research to look at the survival of the virus on paper products, and it doesn't last particularly long, a day or two. Even looking on money too, it inactivates on money. Although mostly that was done with American dollars, so they're paper and ours are plastic. So I don't know if anyone's actually done it on plastic money, but.
Very true. Okay, so let's go to the floor. I have a hand up from the Preville family. Can you unmute your mic, please, and then we'll get your question. Oh. Is it Marcel or Allison from the Preville family?
There we go. I just wasn't sure how to do it. Merci, Anastasia.
Yeah.
I have a question about the masks. So, you know, we're given conflicting information on wearing masks as we go out into the world, whether it's the cloth masks that you've already addressed or the single-use masks. So you're saying that they're not effective for actually preventing you sucking in viruses, but they're effective for me not contaminating others if I happen to be either asymptomatic or symptomatic. Is that correct?
I won't say it completely blocks. So if you're symptomatic and you're wearing a mask, it won't completely block particles from getting out, but it will block most of them. So there won't be very much that can clear the mask, because it'd be trapped on the droplets that are on the inside of your mask. If you are if you're trying to protect yourself and you're wearing a mask, and if someone's standing a couple of feet away from you and they cough and they have virus in their breath, it won't just be on your mask, it'll be on your eyes and it'll be on the rest of your face too. So you're going to be exposed if you're that close to somebody. If you're within 6 feet and they're not wearing a mask and they're infectious, the droplets could hit your face, it could hit your clothes and other things. So the mask isn't really protecting you from them. It keeps someone who's infectious from coughing all over everybody else.
But
Right,
this-
so is a way to then prevent that to, when you do get home after wearing a single-use mask or cloth mask, to wash it immediately and wash your face with soapy water?
Yeah, it's not a bad idea. So one thing, when you put the mask on, you always want to wash your hands before you put the mask on when it's clean. And when you go to take it off, wash your hands, take it off, wash your hands again, because you don't know if it's dirty or not. And then put it in a bag or put it in something. Don't leave it someplace where if the outside does have germs on it, that you're going to pick it up off the surface again later. But yeah, I would, if I was at a protest or something where you're in close quarters with other people and everybody's not wearing masks, I would for sure wash my face when I got home and have a shower, change clothes, probably, even if it, depending on how close the quarters are and how much people are ignoring the idea of wearing a mask.
Okay, and just one final question, if I may. So I've ordered an N95 mask from Britain. It's on its way in August sometime. So I'm told that one will filter out 99.6% of bacteria, viruses, and all other things. So I'm going to be wearing that if I go to shop or those kind of things. Now, how often would I wash, or how would I be protecting myself by putting that on, going out, and then coming back home, taking it off, so it's protecting me while I'm out, but there might be some bacteria on it or virus on it.
Yeah, well, that's why you want to wash your hands when you're taking the mask off. You have to wash your hands before you do it, and then after you do it also, and then that keeps the chances down of infecting yourself. I think right now, Alberta Health Services, I think the only people that are wearing the N95 masks are the people that are working in the ICUs or someplace where there is for sure an aerosol of virus that's generated. I think even people that are working in a room with the patient, if the patient's not on a ventilator, they're not putting a ventilator on or taking it out, I think they don't have to wear an N95 mask, but they also wear a face shield. So that's something you could think about depending on how susceptible you feel like you might be. So if someone was immune compromised, but they had to go out, they could consider getting some kind of face shield to wear, or just wash your face a lot, but the face shield would protect you.
Okay, thank you.
You might, one thing, I don't know whether all the different masks, reusable masks, have different kinds of potential for being re-sterilized and reused, right? So presumably this company offers some kind of guidance on how to clean and decontaminate it. So if it can be put through a washing machine, then that's a perfectly good way of cleaning it and sterilizing it. The other way with the single-use ones that are being sort of reused is that I know some of the health authorities are they just suggest that you carefully take the mask off, put it in a baggie, put it aside, and leave it for a few days. In fact, some people are thinking you have one for every day and you sort of leave it a week and you just kind of keep taking the one from a week before, because the virus will inactivate on storage. So that can give you a very gentle way of cleaning a mask that doesn't involve cleaning.
Okay. We have a few more questions here. We might actually have more questions than we have time. So I'll try to do a quick one first. Follow up on the pools. What about saltwater pools? Do they have the same protection as the chlorine pools?
God, I honestly don't know.
Not a problem there.
Sorry.
That's okay. I'll move to the next question. Dr. Evans mentioned that most CoV-2 is spread through droplets and aerosols, talking, sneezing, coughing, etc. Healthcare workers are told that an N95 mask is not necessary unless performing aerosol generated procedures. Can you comment further, please?
Well, I mean, I think the advice they're getting reflects exactly that observation, that procedures that generate aerosols, large numbers of aerosols, are the ones where you really need to wear a mask for protection. So anything like just simply walking into a room and maybe taking away a pan or something like that, unless you're coming close to the patient, you're probably not going to get infected. The N95s offer a very special level of protection because the 95 refers to the size of the pores which get filtered out. So they're reserved for people who are coming in very close contact to the virus and exposed to high levels of material. Otherwise, they're sort of overkill and they're in short supply. So we're trying to preserve them for the people that really, really need them.
Okay, that's fair. So last two questions I've got is one from Kevin. We're just going a little bit more on the issues of the immunized compromised individuals. So how careful does a person who has a compromised immune system should be versus a person whose immunization is fine?
Well, just to qualify, I'm not an MD, neither of us are MDs, so I don't like to comment too much on individual health situations. But I mean, if you were in that situation, whatever guidance you've had from your doctor or your family doctor would apply to COVID-19 just as much as it would to any other viral infection. So if you've been told your health status would render you susceptible to influenza, for example, or diseases like that, then I think you're probably facing the same kind of challenges and risks with COVID-19. With respect to vaccination, again, that's all very dependent on what the vaccine is, and you really need to talk to your physician about what would be the appropriate vaccine. Hopefully one will come along in the next year or two to provide protection, but that's all very dependent on the specifics of your health status.
Okay, we've got time for one last question. Monica with a K has her hand up. Can we unmute Monica's microphone, please, for her question?
Hi. I am wondering about aerosols produced in a dental setting. And I know that that is one of the places where we are considering that dental workers who are creating aerosol generating procedures should wear N95s or procedure masks with a face shield, something like that. But what I'm interested in is, have you done any research in sort of how long the aerosols take to have like a settle time, like how long it takes for them to settle onto surfaces, how long they remain there? Is there anything like that that you would have done with some of your mechanisms you were looking at?
Yeah, that's a really, really good question. We haven't done it ourselves, but I know there has been a lot of work looking at settle time over the past. Just recently, I was talking to Paul Major, who's the chair of the dental school here, and they are setting up to try and detect virus in the different locations and relative to the patient, as well as the nature of the procedure that's being done. And we're working with a professor in the engineering department who's got methods for air sampling. So I know there's literature on settle time with dental procedures, not specifically related to COVID-19, but looking at other kind of hazards. I know that a lot of work was done around the HIV about 20 years ago.
Right.
If you wanted to send us an email, I could try and put you in contact with Paul and his team, and they could give you some extra information, because it's a huge concern of the Alberta Dental Association right now.
Yeah. Okay. Thank you so much.
Excellent. Well, thank you very much, everyone. Thank you for the great questions. I think there are really a wide range from everything from your general everyday practical things you do to the research itself. So thank you everyone for your great questions. And I'd really like to thank Dr. Hitt and Dr. Evans for spending time with us today, giving us a great presentation and good information and answering all those questions. We did receive a couple of them, like I said, from registrations, and where possible, we will answer them back and I'll send them back the request. I'd also like to thank our facilitators this afternoon, Darlene and Emily, my colleagues in the background that have been monitoring the chat group and helping out with the microphones and helping with and Chrisanne was helping with people getting into the chat group as well. So thank you everyone for all your help. And I'd really like to thank all of you for joining us today. Again, this is a pilot for us. We're learning as we go. We're trying new technology, but we do enjoy being able to bring that scholarship and research information out to the community with you, and we look forward to seeing you again. I will send out an email right away for the next session that will be taking place next week. It will be one of our graduate students dealing in liver research. So that should be a great interest as well. And Dr. Reuse is also this year's 3-minute thesis winner. So that should be a really interesting one. I'll send everyone the information on the next lecture. And with that, I bid everyone a great day. And again, thank you for joining us.